All right, so the next topic associated with this unit is transduction. So remember, there's three parts to signal to a cell receiving a signal and then transmitting that signal into a cell to elicit a response. There's reception, which receives the signal where the ligand binds, which you can see up here. Then there's transduction, which we're going to focus on now, which is how that signal is passed from protein to protein within the cell. And then eventually you get a response at the end, which we'll get to as well. Okay, so transduction, as I said, is just how a signal is passed from protein to protein, or sometimes from protein to non-protein, back to protein again, through this to both spread the signal quickly, to coordinate it, and also to amplify that signal so it really spreads throughout the entire cell, despite there maybe only being one ligand that binds to the cell surface. Okay, so signal transduction pathways. As I said, they are a bunch of interactions between internal proteins that allow for that signal to be passed and allow for coordination, right? That's why there's multiple steps. It doesn't just go ligand binds to receptor and then you get a response. And AP will assess you on that idea. They will sometimes put an answer where the ligand binds to receptor and a response occurs right away. No, it requires many steps in order to both amplify it, but also you want to have many steps to coordinate and regulate that signal, because maybe the ligand that binds is telling your cell to do something, but there's another ligand that bounds separately that tells the cell not to do something or not to do that action. So they kind of cancel each other out. And those different steps in the pathway allow for that type of coordination to occur. So the ways that the signal is passed from protein to protein, right? One is through direct conformational change. That's when one protein will actually interact with another one, changing its shape, activating it. And then once that protein falls off, the protein goes back to being inactive again. Okay. There's cleavage of proteins, which a cleavage means to split. So sometimes a protein or an enzyme will come in and actually remove a part of another protein, turning it into either an active or an inactive form. Okay. Another thing that happens, which is the most common thing that you see, or other than direct conformational change, I would say that this is probably the most common, is the addition or removal of a phosphate group. That's called phosphorylation. When you phosphorylate a protein, usually, not always, but usually you activate that protein. That protein turns on and then it does a job. We'll talk about the mechanisms by which we turn that off in a little bit. And then lastly is the production of what are called second messengers. And second messengers are non-protein, smaller molecules that bind to proteins. They can be produced quickly, that can cause widespread change very fast. So those second messengers will bind to the protein. It'll cause it to have a conformational change, activating that protein or deactivating that protein depending on what that specific pathway shows you. So here's showing you an example of direct conformational change. A G protein activating an enzyme, for example. So you can see down here when that ligand bound, that G protein goes and it binds to this protein right here, activating it. And when it does that, it then produces a molecule. In this case, it's the second messenger, but we'll get to that later. You can see it up here happening as well. This interaction between the G protein and this adenylyl cyclase, as it's called, is an example of a direct conformational change. Then once the phosphate group falls off of the G protein, it will go back to its inactive form and migrate back towards the receptor again. Okay, so that's an example of a direct conformational change. Here's an example of the cleavage of a protein. So you can see here, this is a protein called a zymogen, and it is inactive when this part of the protein is on it. And what happens then is that once this is cleaved off, it exposes the actual active site of the enzyme. That's where the reaction or the chemical reaction can occur. But when that sequence is in there, it masks it. It doesn't allow that to happen. Okay. Here you can hear there's proteolytic cleavage. So a protein will come and cleave this after this protein off. Once this piece goes in and falls off, it migrates in and turns on genes or turns off genes inside of the nucleus. So it just shows you some different examples of how sometimes a protein can be cleaved and can be activated, or sometimes cleaving it deactivates it as well. Okay, so phosphorylation we'll spend a little bit more time on. There are enzymes that are called kinases, and a kinase's job is literally to take an inorganic phosphate groups that they remove from ATP, so they pluck a phosphate group off of ATP, making it ADP, and it will bind them to other proteins. It'll take that phosphate group, put it on a protein. It causes a conformational change in the protein, causing it to either become into an active or an inactive form of that protein. A lot of times that initial phosphorylation sets off what's called a phosphorylation cascade. And in the case of a phosphorylation cascade, what that means is, remember, like kinases will actually activate other kinases through phosphorylation. So there are different types of kinases here. So protein kinase 1 gets activated by a conformational change associated with this active relay molecule here. Now what this kinase does, it removes a phosphate group off of ATP and phosphorylates this kinase, which then is going to go and phosphorylate this inactive protein, making it active, and then that protein is going to do a response now that it's active. Now, there is an enzyme called the phosphatase, which removes that phosphate group, usually deactivating the enzyme. So you want to know that relationship between phosphatases and kinases. Kinases add phosphate groups, phosphatases remove phosphate groups. Usually to activate in the case of phosphorylation and deactivate when it's removed, but the opposite could happen as well. You just have to really look at what the prompt tells you that phosphorylation event does to that specific protein. So phosphatases remove the phosphate groups, kinases add the phosphate groups.
Cells receive signals from outside via receptors. When a ligand binds to its receptor, a signal is transmitted as part of the signaling cascade.
So you can see here's a ligand binding to a receptor, and then you show the transduction.
Signaling cascades are protein kinases. Protein kinases are enzymes that transfer a phosphate group onto a target protein, thereby changing protein function from inactive to active.
So you can see the phosphate group there. Again, sometimes it can deactivate it too, but
Usually use adenosine triphosphate, in short ATP, as a source of phosphate. ATP is composed of adenosine and 3 phosphate groups. Protein kinase binds ATP as well as the target protein and then transfers a phosphate group. The phosphorylated protein and the remaining adenosine diphosphate are subsequently released by protein kinase. Let's take a closer look at the phosphorylated protein. We previously mentioned that phosphorylation leads to the activation of a protein. However, this describes only one out of 3 possible effects. First, the protein may become activated. Second,
most
the
common.
protein may be deactivated.
Also can happen.
The transferred phosphate group can be a signal to other proteins to interact with the target, thereby passing the signal on. Have you ever wondered how the signal is switched off? Another group of enzymes, the phosphatases, are able to remove the phosphate group from the target protein. Phosphorylation is therefore a reversible protein modification that is regulated by a balance of kinase and phosphatase activity. As you can see, signal transduction through the addition of phosphate groups is a powerful mechanism. Accordingly, the enzymes are kinases with the prefix kine, as in kinetics, meaning to move. Protein kinases are driving forces for many cellular processes, and more than 500 different protein kinases have been identified. All kinases are assigned into a fairly complex system of different groups, families, and subfamilies. This classification takes into account both structure and function. Given the large number of kinases, it is impossible to remember them all. However, you may want to keep in mind that they can be broadly classified according to the amino acid residues they phosphorylate. They usually act on one of 3 residues.
I'm going to pause that because that's beyond the scope of the course. That's more college, like even cellular biology. But so as you can see, kinases add phosphate groups, usually to turn on proteins, but to alter their activity in some way. And phosphatases come around and remove them. And they do it almost instantly, so that as long as the signal is always present, a ligand is always there, it's going to constantly keep turning the pathway on. We'll keep reactivating these enzymes by phosphorylation. But as soon as the ligand's gone, the phosphatase removes all of those phosphate groups. But remember, the phosphatase is removing those phosphate groups constantly. But if the signal gets turned on again, then the kinases or the enzymes get rephosphorylated continuously until that ligand is no longer present. The last mechanism I talked about with passing the signals is through second messengers. These are small, non-protein molecules. So these are not proteins. They are water soluble, or they are ions, which obviously are water soluble. And what they do, now I know DAG doesn't look super water soluble, but it can transfer through. It's not water soluble in that sense. It's actually a modified membrane protein, as you can see. Like each of these are molecules or modified versions of molecules we've looked at. This cyclic AMP is modified from ATP. It is a nucleic acid. DAG is a modified lipid. You can see the fatty acid chains here with unsaturated fats with the kinks. This is interesting. So notice this. This is a modified sugar, inositol 1,4,5 triphosphate. But you're like, remember we talked, isn't a sugar supposed to have CHO or just be CHO, right? Oh, in a 1 to 2 to 1 ratio. Look, this one has phosphate groups on it. It's a modified sugar. Okay, there's always exceptions to the rules, but I can tell it's a sugar because of that chair conformation and shape and these other OH groups coming off of it. And then we have calcium, which is just an ion. So cyclic AMP is a very common one you see. It is created by taking ATP and an enzyme called adenylyl cyclase, which gets activated by a G protein, removes 2 of the phosphate groups, and then forms a ring structure with the phosphate group on this one part of the sugar over here, creating cyclic AMP. Cyclic AMP will go around and activate a bunch of other proteins, spreading the signal quickly. So the one G protein activating one adenylyl cyclase will produce hundreds and hundreds of cyclic AMPs before it's shut off. And all of those hundreds of cyclic AMPs will go and bind to 100 proteins, which helps to do a process called amplification, which we'll get into in a little bit. Calcium ions do something similar. So a lot of times calcium is stored in the smooth endoplasmic reticulum. And what you can do is some sort of stimulation can open a ligand-gated ion channel, causing calcium to flow out of the endoplasmic reticulum. And in doing so, cause widespread change very quickly because thousands and thousands, if not millions, of calcium ions can flood the inside of the cell. Well, then once the signal's done, there's a calcium pump that pumps using ATP against the concentration gradient, pumps it back in from low to high back into the smooth endoplasmic reticulum. And the calcium just binds to proteins, right? You can see that down here. So here's an example of a bunch of different second messengers being involved, but also a G protein coupled receptor and a ligand-gated ion channel as well. So the ligand binds to the G protein, it activates, or receptor, it activates the G protein, it slides over, and it activates this enzyme called phospholipase C through a direct conformational change. That will then cleave IP3 and DAG. IP3 will go and bind to, remember that's that modified sugar, to this calcium channel, causing it to open. Then calcium will flow out. Calcium will then bind to different proteins, turning those proteins on or off, causing them to be activated or deactivated, depending. And then that can lead to other proteins being activated, eventually causing a cellular response at the end. So amplification is the process by which, instead of actually, this is not a one-to-one passing of the signal, like was kind of evident, at least in the Rube Goldberg machines we were talking about. Amplification is actually when, like for example, when one ligand binds to one receptor, that actually activates 100 G proteins, not just one like we looked at, it actually activates 100. And each of those goes and activates 100 adenylyl cyclases. Well, each adenylyl cyclase produces 100 cyclic AMPs every time they're activated. And each of those cyclic AMPs goes and activates one protein kinase A. And each of those protein kinase A's actually phosphorylates 10 other phosphorylase kinases. And each of those activates 10 glycogen phosphorylases. And then eventually at the end, it takes the glycogen and it converts it into glucose 1-phosphate, which is our sugar molecules. So we start with one ligand binding to a receptor, of one epinephrine or adrenaline molecule binding to a receptor. And at the end of it, we've created 10 to the 8 individual glucose molecules through that process, through the process of amplification, which is the spreading of the signal throughout the cells. And this makes sense because if we think about what does adrenaline do or epinephrine do, it increases cellular metabolism. It's our stress hormone, our short-term stress hormone. When you are scared or frightened or are playing a sport or whatever, you release adrenaline in order to increase cellular metabolism for survival. We need more ATP or energy when that happens, and that's exactly what the end product is here. So here's showing you that process of amplification. So again, a G protein This is unamplified. So now you can see here, it's just one thing going and activating an enzyme. This is very linear looking. So if this was just like this, there would not be a lot of things. But instead, what we have is this scenario where there's lots of activation. And this is even underestimating here. So one ligand activates, in this case, 4 G proteins. And each of those goes and activates 4 adenylyl cyclases. And many cyclic AMPs are made. So again, this is kind of underestimating it, I would say, in terms of how much is actually produced. But the idea of amplification is still kind of shown here. All right, the last thing we're going to talk about in terms of new content is the response. So what types of responses happen at the end? Well, a lot of times it does something to change the cytoplasmic activities. So enzymes are turned on or off inside of the cell. Or it's going to go and turn a gene on or off, so a new gene product or a protein is made, and as a result, it causes different changes to happen within the cell. All of that happens through ligands binding. There's a lot of different ways to get responses. You can have like this, a ligand binds to a receptor, and then you have these relay molecules with amplification, which is not shown, giving you response 1. Or one ligand can bind and actually can split and give us 2 different responses. Or 2 ligands are required to create 1 response, right? Or one ligand, the same ligand binding to a different receptor, creates a different type of response. There's all different types and ways to get these responses, and you just have to kind of understand about, like, what if we mess up certain aspects or certain parts of this process? What is going to be the impact on the final response at the end? So I did mention about how a lot of times one of the responses is genes get turned on or off. There are proteins, which we're going to get into when we get into genetics, called transcription factors. And those will turn genes on or off. Usually they get activated through these phosphorylation cascades. When we talked about testosterone previously, steroid binding to an internal receptor, it becomes an activated transcription factor. But its job is essentially to go into the nucleus, bind to DNA, and then turn genes on or off depending on the scenario. Okay, so now that we've gone through all the different parts of cell communication, let's talk about some examples that they really like to talk about. One of them is insulin and glucagon, and they're antagonist hormones, meaning they kind of contradict each other. They do opposite things for blood sugar regulation. They're both protein and peptide-based hormones that are produced by your pancreas. They do flow through your bloodstream and bind to cell receptors on the surface of cells because they are peptides. They're not steroids, which can cross the membrane easily. So insulin, when you eat a lot of sugar, will lower blood glucose levels. And glucagon, if you're starving or you haven't eaten in a while or you're doing a lot of activity, will actually raise blood glucose levels. But how do they do this? So if you look here, if you have low blood glucose levels, you have a lot of glucose actually stored in your liver, and in your cells. So glucagon will go to the liver cells, bind to the receptor on the outside, and trigger the release of glucose from those cells and put it into your bloodstream. Essentially like you're eating, but you're just removing stores of sugar from your cells. Insulin does the exact opposite. When the cells when the blood sugar levels are really high, insulin will go in and then trigger cells to uptake glucose and store glucose within them specifically, and most commonly in the liver overall. And the pancreas releases both of these hormones. This is called a negative feedback loop, and we're going to talk about negative feedback loops a lot in this course. If you're in my anatomy class, we've already talked about it, but the best way to define a negative feedback loop is you have a set amount of something, and if you deviate from that amount, you're going to try to always work to bringing it back to that amount. It's a great example of how we maintain homeostasis in our bodies. Remember, homeostasis is that dynamic equilibrium or that balance, despite constant change, attempting to maintain sameness. We always want our blood sugar levels to be between 70 and 100. If it gets too high, we want to bring it down. If it gets too low, we want to bring it back up. Set point, deviate from set point, bring back to set point. So here's showing you what happens at different levels of glucose and concentrations in the blood and how much of each hormone you release. Notice at really low levels of glucose, glucagon secretion is really high because we need to have more blood glucose, so your body's going to work towards getting more glucose into the blood by releasing it from the stores in your cells. In contrast, when glucose levels are really high, insulin levels are going to be high because its job is to take that high level of glucose and then lower it by storing it within the cells and the liver. And that's what this graph is kind of showing you, how they're antagonists of each other. Okay, so how does this relate to what we're talking about? Again, here's showing you the signal transduction pathway of insulin. It is a tyrosine kinase receptor. The insulin molecule binds to it. It will phosphorylate. An autophosphorylation will occur. It activates these proteins, which then triggers the production of this molecule right here, which phosphorylates this, which then triggers GLUT4 transporters, which are facilitated diffusion proteins, which go into the membrane that allow glucose to easily pass through the membrane. Remember, glucose can't cross easily because it's a large polar molecule. This will then bind to the membrane, and it acts as like a trapdoor that alternates and constantly allows glucose to constantly go into the cell, therefore removing it from the blood and lowering blood sugar. Glucagon, on the other hand, that is a G protein linked receptor. Glucagon binds, it activates the G protein, which then goes to adenylyl cyclase, producing cyclic AMP, which activates protein kinase A, which inevitably its goal is to increase the amount of glucose present in the cell from the stored glycogen. Glycogen is a stored form, similar to starch, of long chains of sugar molecules, a polymer of sugar. It will break the glycogen down into individual glucose molecules, and then those glucose molecules will be released from the cell and go into the blood, therefore raising blood sugar levels when they're too low. So what is diabetes? So diabetes is a disease where you do not properly regulate your blood sugar. There are two types. In type 1, that is considered to be a childhood form, though you can get it as an adult too. This is usually when your immune system attacks your pancreas, and your pancreas can no longer make insulin or glucagon. In this case, there is no ligand made. So you cannot regulate blood sugar levels effectively. So the treatment is actually just to inject yourself, test your blood sugar levels, and inject yourself with insulin depending on how many carbs you take in when you eat. There are pumps and artificial pancreas type mechanisms that allow for this to be much more seamless. Back even 10, 15 years ago, you had to really calculate everything. And even 30 years ago, you had to inject yourself directly with insulin and measure it out of vials, which was, there's a lot of possible room for error there, because if you inject too much insulin, it'll actually kill you. It'll put you into a diabetic coma because you lower your blood sugar levels too much. Type 2, though, which is probably the most common type, this is adult onset, and it's usually caused, yes, by genetics a little bit, but by poor diet and being overweight. Now, you can be a type 2 diabetic and not be overweight, and not even have a poor diet if you have genetic predisposition, but the most common cause is obviously a poor diet. And there is insulin in this case. Your pancreas is making insulin. But when it binds to the receptor, something is not communicating properly there. It's not talking. The receptor is not talking to the next step. It's not activating the proteins like it's supposed to. So your body produces more insulin, right? Just to get the same effect. Usually you will be able to maintain blood sugar levels a little bit like this initially, but eventually you cannot. And it can get so bad that your insulin or your glucose levels are high, and you can actually, those high glucose levels can actually mess with your pancreas as well, and you actually become insulin dependent because you don't produce enough insulin after a while on type 2. But this is a treatable in terms of weight loss, but also there's a lot of medications that help with this, including things like metformin and also the GLP-1 medications like Wegovy or Ozempic, which is kind of a buzz drug right now, and it's very effective at treating type 2 diabetes. I'm going to show you this video in class, okay? So I'll show you that in class, actually. So here's a question. What could you test for in the blood to determine if there's the difference between type 1 and type 2 diabetes? Think about that for a second. Okay, what I would do is I would test for two things. One, blood sugar levels. They'd be elevated in both after you eat, and they wouldn't go down. But in a type 2 diabetic, there will be insulin. In fact, sometimes too much insulin present. In a type 1 diabetic, there's going to be absolutely no insulin present in those individuals. So I'm going to relate this quickly to cancer. So growth factors, we talked about these before, are released by cells to tell neighboring cells to replicate, right? We said like growth, if you cut yourself, for example, the cells around those cells will release growth factors and it'll tell cells to replicate. But I don't want all those cells to replicate. Only if the neighbor dies do I want to replace it to replace itself. Now, how does that actually work? There's an example of juxtacrine signaling like we talked about earlier in class. A growth factor will bind to this receptor, and if a cell is not in contact with another cell, you can see this CAM protein is how these two interact with each other to know if there's something called contact inhibition, which this inhibits this process. So if there's no cell here, this receptor activates this protein, which activates this protein, which activates this protein, which then goes and activate RAS, and RAS, through a signal transduction pathway, eventually triggers cell division if it's not in contact. But if CAM is in contact with another CAM here, if these two cells are in contact, this M protein will bind here. And if the M protein's bound, it prevents 3 from binding despite the growth factor being present. And if 3 doesn't bind to the receptor, we never activate RAS. And if RAS doesn't get activated, you get no cell division. So this is a good mechanism for preventing overgrowth of cells. But what if you mutate M, for example? So if M is mutated, and the gene for that codes for the structure of M, and M cannot bind to CAM, despite the fact that they're in contact with each other, what are you going to end up getting? You're going to end up getting 3 binding because M doesn't fit anymore. RAS gets activated despite contact, and then you can start to get the overgrowth of cells in that area. This could be one of the steps that could eventually result in cancer. All right, so let's look at some of these questions and how they appear on the AP Bio exam. So here is showing you a prompt. In eukaryotic microorganisms, the Pho signaling pathway regulates the expression of certain genes. These genes, Pho target genes, encode proteins involved in regulating phosphate homeostasis. When the level of extracellular organic phosphate is high, the transcriptional activator Pho4 is phosphorylated by a complex of 2 proteins, Pho80 and Pho85. Now remember, this is going to be all jargony, right? But what's really happening? In a high phosphate environment, Pho81 does not get activated, so we do not inhibit this process. Pho4 gets phosphorylated by these 2 proteins, Pho80 and Pho85, by removing a phosphate group here. So Pho80 and Pho85 are an example of a kinase because they're phosphorylating Pho4. And then Pho4 will not go and bind to target genes. That's in a high phosphate environment. In a low phosphate environment, Pho81 will actually go and inhibit the activity of Pho80 and Pho85. If they do not phosphorylate Pho4, Pho4 will actually continue doing its job. So this is an example where phosphorylating actually turns it off in the A scenario. Pho4 will then go bind without phosphate group to the target genes. It'll activate Pho mRNAs and it'll produce proteins. So it shows you an example of, again, how this is used. Remember, arrows means activate. This line right here, like a T, means inhibit. So activate, 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 activate. Inhibit up here. Okay? So that's one example. Here's another one. The binding of an extracellular ligand to a G protein coupled receptor in the plasma membrane of a cell triggers intracellular signaling, figure 1A, which is what this is, A, B, and C. So after ligand binding, GTP replaces GDP that is bound to GSA, a subunit of the G protein. This causes GSA or GS alpha to activate other cellular proteins, including adenylyl cyclase that converts ATP to cyclic AMP. And the cyclic AMP activates kinases in figure 1C. In cells that line the small intestine, a CAMP activated protein kinase causes further signaling that ultimately results in the secretion of chloride ions from the cells. Under normal conditions, so again, that's the response, right? The chloride ion release. Under normal conditions, GS-alpha hydrolyzes GTP to GDP, thus inactivating adenylyl cyclase and stopping the signal. So what you can see here, right, ligand binds to receptor, activates G protein, it slides over. You're going to see it go here. It's going to activate this. This is going to produce cyclic AMP. Cyclic AMP is going to activate the protein kinase, and then that's going to eventually trigger chloride release from the cell. So what they're going to do to this is manipulate different proteins. Like, what if there's no ability for the GTP to bind here and GDP stays bound? Well, despite the fact that there's a ligand present, we're never going to activate this adenylyl cyclase, and we're never going to properly activate protein kinase A because we're not producing cyclic AMP. And if we're not doing that, you're not going to get chloride secretion by the cell, and that could be a problem, right? So being able to figure out these just by looking at them and mess with these proteins, it's super daunting. If you've never seen it before, it looks intimidating, but the rules are always the same. Protein activates protein, which produces second messengers, which activate proteins, blah, 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 which you get a response. How is it messed up? Or how does it, maybe it's messed up by either shutting it down or it's messed up by turning it on too much. Here's another one. Here's showing you, this is, let's see, a bacterial infection triggers an active caspase, which then that active caspase goes and activates two interleukins, or this interleukin, and then this one triggers this gastroderm to turn into N-terminal gastroderm, which will then bind to the cell and cause the cell to destroy itself. So this is actually an example of the way that the cell destroys itself if it feels like it's been infected with a bacteria internally that it's now gone haywire. And so it prevents that bacteria from really functioning. It basically kills itself and the bacteria together. Okay, so just another example of these types of things. Here's a capsaicin example. Capsaicin is the spicy stuff that you find in peppers. That's what makes it spicy. When capsaicin binds to this protein here, it's an example of a ligand-gated ion channel. It causes calcium to influx into the cell. That then activates calcineurin, which will dephosphorylate. So that's an example. Calcineurin is an example of a phosphatase. It dephosphorylates NFAT2. Once NFAT2 is now dephosphorylated, it will go into the nucleus, act as a transcription factor, but the presence of it will then turn off the transcription of this, which will turn off the transcript. If this is present, turns off the transcription of this. And if this is present, it blocks or induces apoptosis. But this is the weird thing about the double negative here that confuses people, right? Inhibits. So if NFAT2 is present, what's going to be the impact on apoptosis, right? So if PGOG is present, apoptosis happens. That's easy. But if NFAT2 is present, then no ATF3 will be present. And if there's no ATF3 present, we will not inhibit PGOG. And if we do not inhibit PGOG, there's a lot of PGOG, meaning apoptosis will happen. So if normally this should trigger apoptosis, what if NFAT2 is not present? Then you will not inhibit ATF3. ATF3 will be present in high quantities, and if ATF3 is present in high quantities, it will inhibit PGOG, and you will not get apoptosis. Do you see how this kind of works? These are how you have to read these specific signal transduction pathway mechanisms and models. Another example, the last example, because I like coffee, so we got to talk about it, is caffeine. So caffeine, why does caffeine actually work to make you feel more alert? Why do we take it to give us energy? It has something to do with signaling, but it really has to do with the ligand that binds to the signal. So what causes you to feel sleepy in the first place? So over the course of time, as you burn through ATP throughout the day, one of the side molecules that are produced, which is related to ATP, is called adenosine, which is basically the ATP without the phosphate groups on it, right? Adenosine will go bind to a receptor, and it's a G protein linked receptor, and it will actually cause a signal cascade which results in the slowdown of cellular metabolism, making you feel tired, and it causes you to go to sleep. But interestingly, look at caffeine's shape and compare that to adenosine. Do you see how the base is similar? That's the part that binds to the receptor. So caffeine will bind instead of adenosine. It knocks it out of the way. But caffeine does not cause the same impact. It does not, when it binds to the receptor, it basically gets in the way and you don't have tiredness. You don't decrease cellular metabolism. So it causes you to feel more awake. It's amazing. So caffeine is what's called a competitive antagonist. They're competing with each other, but it kind of does the opposite thing of adenosine. So initially it causes you to feel alert because you're knocking all of that adenosine off. You won't feel sleepy, and it actually increases your heart rate and increases general cellular metabolism. All right, that is everything on cellular communication. If you have any questions, come see me.